My name is Kristen Hicks. I'm the section chief for the Plant Tissue Analysis Lab, Waste Analysis, Solutions, and Media Lab. We're going to talk about soilless media sampling. We call it soilless media because it's important to understand that potting soil is not soil at all. So potting soils should be submitted as media, not soil. For media sampling, there are two different types. You can sample pre-plant or post-plant. Pre-plant sampling is very simple. All you do is take a sample of the media, ideally from the center of the bag or pile, and fill a 1-gallon bag with that media. Do leave some headspace in the top to allow for expansion. And another important rule of thumb is make sure that the media is not dry. It should not be wet and saturated, but it also should not be dry. Dry media will produce inaccurate pH readings. For post-plant sampling, you can sample either destructively or non-destructively. For non-destructive sampling, you remove the plant from its container and you take a small pinch of the media from the middle of the plant and shake it into a pan. You do this from multiple plants, taking only a small amount, enough that it will not damage the plant, from inside of the root ball until you have enough media to submit for sampling. For destructive sampling, you will pull the plant out of the pot. You'll remove as much of the media as you can until you get enough material to submit. Another important tip for post-plant sampling is you want to remove as much of the root material as possible. Remember, we are analyzing the nutrients in the media, not in the plant roots. Once you have enough plant material for a sample, which would be roughly about a standard size Ziploc bag's worth of material, you put it in a plastic bag, label it, and submit it to the agronomic lab. As with plant tissue analysis and solution analysis, be sure to completely fill out the sample information form available online. Media samples, like tissue samples and solution samples, are $5. Fill out the information here with the matching sample ID. Tell us what type of media it is. The codes for that are all on the bottom of the sample information form. And be sure and tell us if you have a corresponding plant sample and if you're experiencing any kind of problems.